Hello and welcome dear students, pharma professionals and learners to the another interesting video, that is Dissolution Studies for Alcohol Induced Dose Dumping. It is a part 3 for this series, and in this mainly the delayed release dosage forms are discussed in detail. Delayed release dosage form are also referred as or called as gastro-resistant dosage forms in Europe. These are done by enteric coating, and the enteric coating is done to protect acid level drugs from degradation in the acidic environment of stomach, to avoid drug release in stomach, or to have a specific release the drug in intestine, then to avoid gastric irritation, then to release the drugs in specific part of intestine for better absorption or local effect. So alcohol induced dose dumping is also required for some of the delayed release formulations, because in delayed release formulation the release of the dosage form is modified and there is a lag time in the release of the formulation. That's why for delayed release formulations or gastro resistant formulations, the dissolution is performed in 0.1 normal acid for 1 hour or 2 hour, followed by the basic pH. So alcohol induced dose dumping is required for some modified formulations like extended release, controlled release, prolonged release, and also for delayed release formulations or enteric coated formulations, as the release in DR formulation is controlled with the use of polymers, and as loss of DR design may lead to dose dumping and release of dose in stomach instead of intestine. That means the delayed release target is hampered due to may hamper due to presence of alcohol in the GI. This dose dumping may lead to loss of activity or formation of impurities for the drugs. Those are unstable in acidic or alcoholic environment. So to support for delayed release label claim, alcohol dose dumping study is required. Also, the alcohol induced dose dumping studies are required for comparative dissolution of test product or generic product along with the RLD, and also for supporting data for lower strengths for biowaivers. So delayed release formulations are required to be studied for alcohol induced dose dumping. Now I have some examples and ready references from which we can be able to understand the study which are required for alcohol induced dose dumping for the delayed release dosage forms. And for that I have considered some examples like posaconazole delayed release tablets, octreotide acetate delayed release capsules, duloxetine delayed release capsules, and budesonide delayed release capsules. First of all, we will consider posaconazole delayed release tablets. This is an antifungal formulation, and enteric polymer used in this formulation is hypromellose acetate succinate. So the hypromellose acetate succinate has limited solubility in alcohol. So alcohol dose dumping study may be asked due to API solubility or to support delayed release design. The testing conditions are 900 ml 0.1 normal HCl, USP apparatus 2, that is paddle apparatus at 50 rpm with and without the alcohol. So without alcohol, dissolution will be performed only in 0.1 normal HCl, and with alcohol, concentrations of alcohol to be taken as 5%, 20% and 40%. So these alcohol concentrations are as per USP or US guidelines. Instead of this, we can go for Europe requirements also. So 12 units are required to be tested in the proposed method with 0.1 normal HCl, and data to be collected every 15 minutes for total of 2 hours. This is required to support the delayed release design, or maybe due to the solubility of API and the polymer. Then another example is octreotide acetate capsules. These are delayed release capsules, and enteric polymer used is methacrylic acid ethyl ethacrylate copolymer with ratio of 1:1, and the alcohol dose dumping may be asked due to polymer solubility and delayed release design. So here the dissolution test is to be performed in 900 ml 0.1 normal HCl USP apparatus to paddle 50 rpm with the different concentration of alcohol like 5%, 20% and 40%. Here also the requirement of data is there, similar to the previous example of posaconazole tablets. 12 units data to be generated, and every 15 minutes data to be collected for total of 2 hours. So 2 hours is considered considering the gastric emptying time. Another is duloxetine delayed release capsules or gastro-resistant capsules. This is a very important example and very specific example considering the delayed release formulation. So enteric polymer used in this formulation is hypromellose acetate succinate, and ADD is required due to API instability in the acidic pH and delayed release design of the formulations. So for these examples I have considered product specific guideline as per the US FDA and the dissolution criteria. 10% and 40% is not required by the EMA. So 12 units are required to be studied till 2 hours and data to be collected for every 15 minutes. See here the data requirement is for dissolution data and also for the percentage of breakdown products. These are the impurities or degradation products, 1 naphthol and 1 for rearrangement compound as each time point. Mean value, standard deviation, coefficient of variation, that is percent CV, and the plots of the percent release profile of duloxetine, 1 naphthol and 1 for rearrangement compound over the 2 hour period is required to be generated. So along with the dissolution, the impurity generation data is also required. Then the test 2, in vitro 6-hour stability testing. This is somewhat different requirement for this formulation due to concern that the patients with delayed gastric emptying time may experience release of duloxetine in the stomach, leading to the formation of 1 naphthol, a toxic compound. You should conduct comparative in vitro stability testing on all strength of the test and RLD products using 12 units using the following method. Test method is 0.1 normal HCl 1000 ml without alcohol, USP apparatus basket at 100 rpm for 6 hours. So this is the requirement that the test formulation is required to be studied for impurity generation or degradation product generation in the gastric media, that is 0.1 normal media, for 6 hour time, considering the patient population which may have the delayed gastric emptying. So data required here is individual dissolution data for duloxetine, percent of breakdown product 1 naphthol and 1 for rearrangement compound as each time point, mean values, standard deviations, coefficient of variation, and plot of the percent release profile for duloxetine, 1 naphthol and 1 for rearrangement compound over 6 hours period. So this is very interesting example for duloxetine release capsules, where along with the alcohol dose dumping, the requirement is there for impurity generation in the dissolution media till 6 hours. Another example is budesonide delayed release capsules. Enteric polymer used in the formulation is methacrylic acid, ethyl ethacrylate copolymer 1:1 and 1:2. So the polymer with two different ratios in combinations are used for the enteric coating, and ADD may be asked due to the polymer solubility and delayed release or targeted release site in the GIT. So for this product, there is a claim of delayed release. So specific site is there in the GIT where the product will release the contents. So for that, the alcohol induced dose dumping dissolution studies are required for this product. And interestingly, the media is 0.1 normal HCl 900 ml USP apparatus 2 paddle, and the speed is 100 rpm. See, the speed of paddle is generally 50 rpm, but here the apparatus 2 is used at 100 rpm with and without alcohol. Alcohol concentrations are 5%, 20% and 40%, and the requirement is similar to the other products, like 12 unit data to be generated with the proposed method, and data is collected every 15 minutes for total of 2 hours. So this is regarding the delayed release formulations. Now you might be clear that why these formulations are tested for alcohol induced dose dumping. See, mainly the dose dumping is required so that the formulation is proved to have delayed release profile, and the delayed release profile or the formulation design is maintained in the alcoholic media. So for this for this video I have referred USFDA guidelines and the product specific guidances available. So every time the MR formulation or DR formulation is taken, it is not required to be crushed. So don't crush or chew the DI formulations and promote the same. Then stay tuned to this channel for more informative videos and interview preparations. Also promote to avoid alcoholic drinks. Thank you for for watching this video, and do like, share, subscribe to Pharma Learning in-depth channel. Thank you.